All right. Well, good morning, everyone. Thank you for joining us today in the seventh webinar in the ASL webinar series. My name is Josephine Lembong. I am the Senior Manager of Science and Industry Affairs at the Alliance for Regenerative Medicine. And today's webinar is on the topic of cell collection and cellular starting materials, and will be delivered by the contributors of Chapter 5 from Terumo, who I will introduce in a few minutes. So for those who are not familiar, ARM is the leading international advocacy organization championing the benefits of engineered cell therapies and genetic medicines for patients, healthcare systems, and society. We as a community built the future of medicine by convening the sector, facilitating influential exchanges on policies and practices, and advancing the narrative with data and analysis. We actively engage key stakeholders to enable the development of advanced therapies and to modernize healthcare systems so that patients benefit from the durable, potential curative treatments. As the global voice of the sector, we represent more than 475 members globally, including emerging and established biotechnology companies, academic and medical research institutions, as well as patient organizations. So I'm sure everyone here knows that manufacturing for cell and gene therapy is a complex process and is often a rate-limiting step in the commercialization of these therapies. This industry overall is in the process of rapidly scaling up. There are more phase 3 studies and more commercial product approvals anticipated. But products are being held at the BLA stage due to some CMC issues. So we see that the advances are being made in the manufacturing technologies and the experience in the workforce overall are improving. However, standards and best practices in the field are not advancing as quickly, and this is the area where ARM focuses our efforts in to further advance the process understanding in the industry to facilitate product development and commercialization. ASL is a project that ARM undertook starting several years ago after feedback from our members about the CMC issues that the field overall as a whole is experiencing. ASL is a best practices document which incorporates collaborative case studies presenting the implementation of QBD principles in cell-based therapies. We chose to focus on immune cell therapy as a case study, and in particular, we further focused this document on autologous CAR-T, given the recent approvals and also the number of products that are available to patients in the market. So these efforts follow the success of AGene, the sister project of ASL, which focuses on AAV vectors, and also AMAP, which was instrumental in allowing advances to occur in the monoclonal antibody industry. So we're very fortunate at ARM to have a large number of subject matter experts from our member companies. We have many expert contributors for this project from more than 30 ARM companies. And this project is also supported by Nimble, who generously provided funding, which allowed us to employ technical writers for this document. The document and the webinars are intended to benefit the entire field, so it is available as open access on our website at the link provided here. This is what the contents of the document look like. And as you can see, it covers quite a big scope from regulatory consideration, comparability, standards, the foundational principles of QBD, starting materials, to the control strategy, and last but not least, facility design. Today's talk will cover the elements of cellular starting materials laid out in Chapter 5. The webinar today is the 7th out of a 9-part webinar series for the ASL educational rollout. The schedules for the webinars are posted in this link. We hope that ASL, both the document and the webinar series, can serve as an educational tool to this community. And we hope that the document can be used by the product developers or educators at universities. And we also have the aspirational goal that this will be used to help educate regulators. And before we move on, just one housekeeping item. You can use the Q&A function to ask questions, and we'll try to answer as many as we can at the end of the talk. And if we didn't get to your questions, we will compile them and post a response on the ASL webinar website. And with that, I would like to introduce our expert speakers for today's webinar. So Dr. Dalip Sethi is an innovative and transformational scientific leader. He currently serves as the scientific lead for Terumo BCT cell therapy technologies portfolio. He holds a doctorate and conducted postdoctoral studies at Thomas Jefferson University School of Medicine. In his postdoc, Dalip focused on the development of cancer gene-specific RNA and DNA analogs targeted against cancer genes and signal transduction pathway for use as cancer diagnostics and therapeutics. Throughout his career in the industry, Dalip has been engaged in developing technologies and methods for use in cell therapy applications. He has authored multiple scientific publications and is a co-inventor on several patents and patent applications. Dalip is also an ISCT member and participates in committees focused on cold chains, particulates, and process analytical technologies. Dr. Erin Goodhue is a national leader in transfusion medicine and therapeutic apheresis with over 15 years of clinical experience. She has led and collaborated on multiple national apheresis and transfusion medicine trials, particularly in pediatric patients, and authored many journal articles and book chapters. In her current position as Director of Clinical Research and Development at Terumo, Dr. Goodhue provides strategic clinical direction for new and novel Terumo products. She also leads a team of clinical and laboratory scientists who collaboratively evaluate, prioritize, and implement developed strategy, including clinical trial protocol development and implementation, as well as post-market studies. And Elizabeth Valdez is a registered nurse with 11 years experience in the field of apheresis. She is currently the Integration Manager, Therapeutic Services at Terumo BCT, where she serves to enhance customer experience by providing services that go beyond medical devices. She graduated from Regis University in Denver, Colorado with her BSN, and in 2018, she completed her MSN specializing in nursing education. Former positions, Liz had our global trainer for Terumo BCT and apheresis operation associate manager at Novartis. These positions, paired with her academic credentials, have shaped her dedication to expanding apheresis education for device operators and enabling network opportunities within the unique subspecialty. Dalip, Erin, and Liz, thank you again very much for taking the time out of your busy days to be here today. And I will turn it over to you to start your presentation.
Thank you, Josephine. And I will ask you when you can see my screen, just give me a yes, so we know that we are sharing.
Yeah, we can see it.
Cool, thank you so much. So first of all, I would like to thank ARM, Nimble, and the organizer for this webinar, keeping this education going forward. It's a critical piece in the overall cell therapy manufacturing. The starting materials are a key. So thank you for that. And thank you everyone for joining us today, taking time out of your busy schedule to be with us today. So I'm very grateful and thank you for being here. So before we talk about this chapter, cell collection and starting material, I want to give a little bit of brief overview of Terumo BCT. The Terumo BCT, Terumo Blood and Cell Technologies, we design and manufacture products that collect and process blood and cells from donors and patients that help deliver effective therapies. We are headquartered in Lakewood, Colorado, operate globally, and will be celebrating our 60th anniversary next year. We pride ourselves as we invest in innovation and services to advance automation, expanding access, and provide scalable solutions to help bring critical therapies to patients. Our customers involve blood centers, hospital, therapeutic apheresis, clinics, cell collection and processing organizations, researchers, and private medical practices. So the Terumo BCT organization goes across the span of when you think about cell therapy manufacturing, from cell collection to manufacturing. We provide instrumentation and devices for manufacturing. So we pride ourselves in bringing this innovative products to help therapy developers develop their therapies. So that was a quick, brief introduction, and I'm very happy to be here with my fellow experts and panelists. I'm so lucky that they are my colleagues. I work with them on a daily basis. So with that, I'm going to pass to Liz, and I'm going to be managing the slides. So Liz can tell me when to go next. But with that, over to you, Liz.
Thank you, Dalip. I appreciate the introduction. Yeah, so go ahead and we can forward and we'll dive into the webinar. As Josephine had mentioned, sorry, I wanted to go back. As Josephine had mentioned, this webinar is really just a high-level overview of the chapter that we did in the ACell book. So if you wanted further reference, you can go to that ACell book and get some more details on what we're going to be talking about today. Next slide. This is the intended use for Spectra Optia apheresis system. I'm not going to go over this in detail. You can read that at your leisure. You're going to be given a copy of this webinar, so you can read it at that point. Next slide. So before we kind of move forward, I always like to start with good working definitions, just so we level the playing field so everybody knows what we're talking about when we speak about apheresis. So apheresis is a medical procedure that involves the removal of whole blood from the patient or donor and separating out those components of the whole blood, and then taking whatever whole blood component you want to keep and returning the rest back to the patient. An example of an apheresis procedure is the leukapheresis procedure, and that's going to be the focus of the webinar today. In the leukapheresis procedure, you're isolating the MNC cells from the whole blood in the patient, you're removing those up into the collection bag, and then you're returning the red cells in the plasma back to the patient or donor. Next slide, please. So just some kind of fun information about the Spectra Optia. It's manufactured, as Dalip had mentioned, by Terumo Blood and Cell Technologies. They have been involved in the apheresis cell collection space for over 40 years. When we look at the use of Spectra Optia in some of our markets, specifically North American market, about 94% of MNC collections that are done in that market are done on the Spectra Optia. When we look out globally, it's about 67% of the MNC collections are done on the Spectra Optia. Another kind of fun fact about Spectra Optia is it can perform additional types of apheresis procedures. That's kind of important to know because a lot of the apheresis centers are busy with all types of different collections. So it's just kind of something to keep in mind when we're talking about scheduling of cell collection procedures. Just realizing that there's other procedures that are going on in that particular clinic space. Next slide. This illustration is really just a high-level overview of what is actually happening during an apheresis procedure. And so when we look here at the patient in the middle, we can see, most importantly, that when you're doing a leukapheresis procedure, you have to have 2 points of vascular access. So when we look over here on this side, we can see that the whole blood is being removed from the patient via a blood pump, and it's mixed with an anticoagulant before it is entered or during the time that it's pulled into the tubing set that's being used. That whole blood is being pumped into the centrifuge where the cell separation or the whole blood component separation is occurring. In this example, we are removing the MNCs from that separated whole blood and putting that into a collection bag. And then we are taking the red cells in the plasma and removing that back into the patient, which is where that second point of vascular access is needed. I'm not going to go too deep into the vascular access. It is an important point to remember when you're talking about apheresis because Dr. Goodhue will be talking about that during her presentation. Next slide. Okay, I'm going to have you forward one more time because I kind of did this slide backwards. So when I think about talking about, well, what's going on in that centrifuge? When you are pumping that whole blood down into that centrifuge, what is exactly happening down in there? And so this illustration here is meant to represent part of the tubing set that's loaded on to the centrifuge, and it's called the channel. So what is happening is the whole blood is being routed down into the centrifuge chamber area. Here, the anticoagulated whole blood. So this channel is spinning on top of the centrifuge. So under centrifugal force, we see that the whole blood is being separated into its individual components. Down here at the bottom, this neat little plastic piece, I love this plastic piece, we'll talk about it a little bit more, it's called the connector. And this is where we see evidence of the separation of the whole blood. And then you can see that we have these various ports coming off of the connector. And really what those ports are designed to do is to route those components back to where they need to go. Okay. So when we see that the, thank you, Dalip, that's helpful.
I'm trying.
What is she talking about? So when we can see that the RBC and the plasma ports, those are directing those components to go back to the patient during the procedure. Now we can see the collect port here, which is where the MNC fraction is at. That is being pumped out of the connector up into the collection bag. Okay. When we think about how cells are separated inside the connector, again, it is specific gravity separation. So if we kind of think about whole blood, and these are very general statements that I'm making here. When we think about the whole blood of a patient or a donor, typically speaking, about 45% of that patient's total blood volume is packed cells. Now, I know that there are probably some apheresis nurses that are on this call that are saying, that's not what I experienced. Their counts are usually, their hematocrits are usually a little bit lower. And I would agree with you, especially in the CAR T patient population. So these are just generalizations. When we think about the liquid portion of whole blood, about 55% of that is plasma. And then when we talk about the buffy coat, about 1% of the patient's total blood volume or donors is buffy coat or MNCs. If we were to drill down even a little bit further into that buffy coat separation, we can see that we have some extremes, with granulocytes being the heavier of the, or the more dense cell. Those cells usually kind of separate down into the red cell layer. And then we have the platelets, which are the lightest of the MNCs, and those tend to float up into the plasma area or at the top of the mononuclear cell layer. So that center point, the lymphocytes and the monocytes, are really what we're targeting during this procedure. And what I really want to mention here is I get a lot of questions about specific cells. Are we able to go in and target, for example, just an NK cell? And I would say that that does not happen, because NK cells kind of fall into that specific gravity of the lymphocyte and monocyte. So really what you're targeting is that whole cell population in and of itself. Okay, next slide, please. So I mentioned the connector, it's like where all the magic happens. I really loved the design of this plastic piece. But one thing I wanted to call your attention to was that term, the interface. And you're going to hear that a lot in the apheresis industry. And what we mean by the interface is the separation of the cellular components of whole blood from the liquid portion of whole blood. Okay. And Optia is always trying to maintain that interface, that is that separation of those cells at this collect port. And how Optia does that is through a unique camera system called the AIM system, the Automated Interface Management System. So Optia is always fine-tuning that plasma pump to make sure that the cells, the cellular portion of that whole blood, is maintained at that collect port so we can pump those cells off into a collection bag. Pretty uniquely made. And then you can see the other two ports we discussed are the red cell port and the plasma port, which are directing the flow of those components back to the patient because we don't want to keep those. Okay, next slide. Another concept I wanted to introduce would be the concept of packing factor. So really what packing factor is the speed of the centrifuge in relation to how fast you're bringing in cells into the device. I think there's a background picture, Dalip, if you wanted to forward one more. Yeah, there we go. That's perfect. So if we see this picture over here, that would be on my left, probably your right, looking at it. When we talk about high packing factor, what that means is the centrifuge is spinning at a high rate. When we spin the centrifuge at a high rate, what that tends to do is it smashes, and I know that's probably not a good technical term, but it's how I think about it. It takes all that cell component of the whole blood and it smashes it together, right? So we don't have those distinct layers. We have the red cells, MNCs, and platelets kind of all packed together. Now, when we talk about a slow spin or a low packing factor, what is happening is the centrifuge isn't spinning as fast. And so what that does is the cell components of the blood tend to be a little bit more looser, if you will. And what that does is that allows us to move that MNC layer away from the red cells, so we're able to go in and pump those MNCs out into the collection bag. And also what it does to the platelets is it suspends those platelets up into the plasma. So we're not collecting those and we're sending those back to the patient. We're minimizing the collection of platelets. Okay, next slide, please. So there are 2 cell collection protocols available on Spectra Optia. And Spectra Optia was introduced into the North American market. It was like in the mid 2000s. I don't exactly have the date, but it was in the mid 2000s. The first protocol that was available on Spectra Optia was the MNC collection protocol. It was the only one that was available on that particular device at the time. The MNC protocol is pretty interesting because it's called a dual stage separation technology. And what do I mean by that? First, the whole blood is separated into its components. Once that cellular portion of that blood is separated into its components, that cellular portion is moved into a secondary chamber where the cells are now separated by size. So they take advantage of the size of the cell, with platelets being the smallest of the cellular portion of the blood, with red cells being the next smaller. And then we have white cells that are the bigger type of the cells. So when inside that chamber, we have this countercurrent flow of plasma coming through that chamber that allows those cells to separate out by size. So if you can think about it, you're accumulating those MNCs into that chamber. When that chamber is full, then you're going to go into a collection cycle. Okay. We mentioned that it's suited well for small patients. And the reason we say that is because of a concept called the extracorporeal volume. Extracorporeal volume, I'm just going to give you a brief definition of that, is the volume of blood outside of a patient's body at any given time. So if you can imagine, the extracorporeal volume on this particular kit or tubing set is 147 mils. If you had a patient that was really small, 500 mil total blood volume, the removal of 147 mils is a lot of blood volume for them. But there are interventions that we do have that Dr. Goodhue is going to go over to help the patient be able to tolerate that procedure. The other protocol that came out, I want to say about 2015, was the continuous mononuclear cell collection. Now, I would say in our North American market, probably about 80 to 85% of cell collection facilities are using the CMNC protocol. And how CMNC differs from MNC is it's a specific gravity separation with the removal of the MNC at a prescribed mil per minute rate during the entire procedure. So it's not kind of like this batch separation. You are continuously moving those MNCs up into the collection bag during the entire procedure. It utilizes that soft spin or slow spin that I had mentioned. We're suspending platelets up into the plasma. We're separating the MNCs away from the red cells. So the collect port is moving those cells up into the collection bag. You can see the extracorporeal volume is big. There's a lot of blood that's inside of that channel at any given time. So that becomes something you need to think about when you're talking about taking care of smaller patients. And then we have some operator controls. They're fairly similar between the two with your inlet flow rate, meaning how fast are you bringing the cells in? And that really, what limits you there on your inlet flow rate is your choice of vascular access. Collection preference is really how deep inside the MNC layer are you collecting. We're going to go over that a little bit more when we talk about optimization. And then the collect flow rate on CMNC is how fast are you moving those cells out of that connector up into the collection bag. Okay, next slide. This illustration here is really to demonstrate the differences in the kits. We kind of went over this in the previous slide, but one thing I just want to point out is the MNC collection. You can see that connector where you have specific gravity separation. Those cells are then moved into that secondary chamber where it's allowing those MNCs to accumulate because now we are separating out by size versus the CMNC kit that is on the other side where we are separating solely by specific gravity and moving that MNC layer up into the collection bag. So there is a difference in the kit. Next slide, please. I love this slide. I love this slide because it really kind of points out that there are differences in that buffy coat separation, depending on the patient population you're talking about. And I think it just illustrates that you really have to consider, you know, is this a mobilized patient? Is this not a mobilized patient? Because the buffy coat in a non-mobilized patient, particularly a CAR T patient, is small, it's thin. Your window for collecting is a lot, your target, I should say, not your window, is a lot thinner, right? So that you have to really kind of think about your optimization techniques in this patient population. When you compare that to a mobilized patient, the buffy coat is pretty, you know, like it's beefy, right? Like you're like, oh yeah, I can go in and grab all of those cells for the most part. So there are some differences that need to be considered depending on the patient population. And I will end it there, and I'm going to hand it over to Dr. Goodhue. Thank you.
Thanks, Liz. That's fantastic. So just looking at the your basic kind of collection efficiencies, when you're collecting lymphocytes or using CMC to collect more of your stem cells, about what you can expect and what has been shown to be expected in the literature. Obviously, many different things, as Liz discussed, and I'll continue to discuss, as will Dr. Sethi, go into this, but roughly around 60% collection efficiency for lymphocytes and for CMC can be pretty much expected. Lymphocytes representing the MNC here. So something you can look at, obviously, with your own patient population, donor population, as well as your own operators, and it's something super important that you can track yourself. Next slide, please, Dr. Sethi. So there's a lot of considerations that Liz kind of already alluded to. And the first one I'm going to talk about is extracorporeal volume. Next slide, please. I've also worked predominantly with pediatric patients, as I know Liz has as well. So it's something, or small or anemic patients, as I'm sure many of you have or will, especially in the stem cell CAR T space. So being very aware of this is vital, and knowing the extracorporeal volume of your disposable, your device is a vital importance. So you can make sure even vulnerable patients can have a very successful and safe stem cell or immune effector cell collection process. So per FDA standards and other areas of the world, your extracorporeal volume for your patient or donor should not exceed 10 to 15% of the patient's total blood volume. I'm sure many of you have collected very small patients or considering doing that. I myself, I think my smallest was 3 to 4 kilograms, and that's several hundred milliliters of blood volume. So you need to know what your exchange set on Optia is, what your disposable kit for your CMNC and MNC is, and what the maximums are. And we kind of have a nice table here to kind of show that for you. And as you can see, for CMNC, typical ECV, extracorporeal volume, is around 253 milliliters. And as I said, very small patients or very anemic patients, that can be challenging. But we have some things you can consider to still help your small volume patients and your anemic patients have a very successful collection. Next slide, please. One thing we have implemented in Optia, which we did not have in our previous, allows you and walks you through in this nice interface how to set a custom prime for your patients. And what Optia does is it recommends it if you are going to exceed that 10 to 15% of your patient's total blood volume on that specific disposable set, CMNC or MNC. And this will help trigger that. In addition, if your patient is anemic, as many of them are, just knowing the disease states that we're helping treat here. So the team should look at this and consider collaboratively when they could do a blood prime, most likely potentially a red cell prime, to help mitigate this volume shift and this red cell mass shift for their patient. Next slide, please. And Optia helps you kind of get there and start thinking about that to open those conversations together. So during the procedure, the saline in the tubing set is replaced with blood, so that the blood, that additional blood to prime the set, is not pulled from the patient, but from a unit of appropriately cross-matched red blood cells or modified whole blood. When the tubing is attached to the patient's vascular access, the procedure is started, that donated blood fills the set and prevents that extra volume from being taken. So that mitigates that initial first volume shift and sets the patient up very well for a very stable procedure, which is what you want, to keep that interface very stable, to keep that patient very stable, so you can get the most efficient collection as possible, as Liz has discussed. Then those red cells from the red blood cell unit will then go into a waste bag during the procedure. Next slide, please. Next thing that is incredibly important, besides making sure your donor or your patient has the appropriate extracorporeal volume for your tubing set and mitigating that with a potential prime, is to look at anticoagulation. So if the circuit is not appropriately anticoagulated, you don't get that great stable interface that Liz was talking about to get the most efficient collection on the Optia as possible. Next slide, please. And this begins at the very moment that the needle hits the vein. And what Optia uses is an anticoagulant called citrate, which works by binding calcium in the body. And as we know, a small percent of calcium is in the plasma. The majority of it is in our bones, but within the plasma, most of it is bound to plasma proteins. And calcium is an incredibly important cofactor in the anticoagulation system. Next slide, please. So what we do with our anticoagulant of citrate is bind up all that calcium in the circuit to ensure that we've got no clotting and can reach that stable interface. So, and it starts right as soon as that, that needle hits that vein. So our ACDA is what Optia is approved to use with. Love it clinically myself in patients because it has a very short half-life. It is metabolized for the liver. So you do need to consider in patients with immature livers or patients who are having some liver dysfunction. You have to monitor them a little bit more closely. And it anticoagulates the circuit, not the patient and the donor. And if a small amount does reach the patient and the donor, with a healthy liver, it is metabolized very quickly. It binds that calcium and it inhibits activation of our calcium-dependent coagulation factors, which I believe I had to memorize at some point for boards. And it also has another effect of lowering the pH of the blood, which prevents platelet aggregation and keeps those platelets in suspension. As many of us know, platelets love to activate when they see plastic. There's a lot of plastic in the entire disposable. So the citrate has a nice dual effect here by binding the calcium-dependent coag factors and keeping those platelets from aggregating. And then it's just nice with the dextrose and the ACDA, which helps nourish the collected cells until we can get them to the appropriate bag. So it does not fully anticoagulate the patient, very safe. It anticoagulates that circuit so you can get that really fantastic stable interface. Next slide, please. Then the rate at which the anticoagulant infuses is important, especially with something you need to talk to your patients about to make sure they're aware of potential risks. If their calcium does drop a little bit, they can see some numbness and tingling in their lips or their mouth, or they can feel nauseous. These are things that the clinical team has to be aware of when they're consenting the patient or the donor. But on Optia, what we calculate the rate of AC infusion is the milliliters per minute per liter of the total blood volume. And this is called our citrate dose rate. We can configure it on Optia to 0.8 to 1.2 mils per minute per liter total blood volume for all procedures. So you will be able to, on your Optia, based on your patient, how your patient, your donor is doing, work within that citrate dose rate to ensure adequate anticoagulation of the circuit and then comfort of your patient. The default value is 0.8 mils per min per liter automatically. Next slide, please. Then defining the inlet for AC ratio is also incredibly important to keep that circuit very well anticoagulated for that separation. It is 1 part AC and the remaining parts whole blood. And it represents basically that tubing anticoagulation. So always during a procedure, you should be looking at your disposable, monitoring everything to make sure flow has been adequate. Even sometimes before the Optia tells you there's an issue, you may potentially see something happening within the circuit. So the inlet AC ratio is a number that represents the relationship between the rotations of the inlet pump and that AC pump to keep that circuit very wonderfully anticoagulated. Next slide, please. If there's ineffective anticoagulation, as we can see here, it's some pictures in real time of the manifold of the collection port, you can see that there isn't going to be great separation, and then you'll get a lower collection efficiency, and your procedure could potentially take even longer, which is very difficult, not only for the patient and donor, but also for the collection team, and can be very frustrating, especially in some cases, if you're mobilizing a patient, you really, or donor, you only have a certain window of time. Next slide, please. So my last thing that I'm going to discuss that's incredibly important is vascular access. This can set the tone for an entire procedure and is something that needs to be discussed, again, collaboratively with a patient, even the family, and the clinical team and the apheresis team as well. Next slide, please. There are many things that determine vascular access. And yes, Dr. Sethi, you can just walk through this. I apologize, I forgot. I made it. Thank you, sir. Oh, thank you. Acuity, number and frequency of treatments, duration of treatment, the patient's vascular anatomy. We're all very different in that case. There are some similarities, but there are some uniquenesses: mobility, hygiene, comfort, age, and where the treatment is happening, the collection is happening. Is it inpatient versus outpatient? Experience, comfort levels of providers. Certain providers have certain favorite vascular access devices, catheters, IVs that they like to use, ultrasound machines. So usually for an outpatient treatment, there tends to be emphasis on more peripheral IVs. Inpatient treatments, especially those who are potentially critically ill and have difficult vascular access, can use potentially central catheter. But again, it is paramount to know what the comfort level of the providers are at your site, and to have discussions very collaboratively together, especially to make sure the patient and the family and the donor even are very well informed of their choices. The nice thing is Optia is very flexible and can get you a really good, efficient collection with multiple vascular access options. Next slide, please. This is a nice paper that was just published a little while ago that looked at the different types of vascular access, frequency of use in several different locations. And as you can see, there is kind of a tendency for more tunneled or central catheters, but we're starting to see a rise in peripheral access, especially if the duration of the procedure is going to be relatively short and the patient feels comfortable and can tolerate that. Next slide, please. So the number of alarms can be really high on the Optia when your vascular access is inadequate. And trust me, when I tell you, when I'm in clinic, some days I feel like I'm spending a third up to even half of my time, and my nurses as well, when dealing with issues in vascular access, because that can really make or break a procedure. Can you go back, Dalip, please? And as you can see here, this can lead to lower collection efficiencies. And so that's something that needs to be set and worked on together as a team, including the patient and the family upfront, to make sure you get the best collection possible. Next slide, please. So it's a physician and patient decision, apheresis team as well. What is available, what is comfortable, and guidance to customer practices. Definitely needs a cross-functional team so that you can get the best collection possible. Next slide, please. Now I turn it over to Dr. Sethi, who will take you through the amazing donor considerations for this type of therapy. Thank you, Dalip.
Thanks, Dr. Goodhue. Thanks, Erin. Thanks, Liz. It's been exciting so far. So we also covered in this chapter some donor consideration on allogeneic cell therapy products. As we know, in the CAR-T space, a lot of companies have started moving towards allogeneic cell therapies, where a single donor can produce drug for multiple patients, where compared to autologous, is the same patient who's giving the cells and is receiving the therapy. So we have covered in detail in the chapter, so I'm not going to go to every single thing here. What we put in this main topic, there's consideration. So when you're thinking about allogeneic therapies versus autologous, one thing is common: the manufacturing has to be robust. And when we think about the safety of these drugs, we think about adventitious reagents like viruses, bacteria, mycoplasma, and other things that can come. And there are multiple ways that they can be introduced into the manufacturing. It could be the starting material. It could be the products that have been used for manufacturing. It could be the handling of the cells during the manufacturing. So as a developer or as a provider, you have to be aware of all those things and put controls in the place. So when we think about starting material qualification, in this chapter, we referenced a 2020 workshop that was done, and we talked about where there is a safety consideration where you think about all the testing that needs to be done from a safety perspective, testing for different viruses, testing for different diseases. And then we talked about the qualification of the material in terms of how many MNCs you have, what is the viability, how many CD3. A particular developer can think about further testing that they want to do for starting material qualification. They can look at further phenotype of how much percentage of NK cells are present, what is the percentage of Tregs, for example. So it will depend, but the basic qualification is definitely from the safety perspective, and then the TNC, and for CAR T, the CD3 is an important parameter along with viability. We also covered extensively in this chapter, and please have a look. We talked about the donor screening and testing. So donor screening and testing may not be regulated in terms of autologous, but it's very regulated in terms of allogeneic cell therapy. So it's very important to test for adventitious reagents, like we think about HIV, type 1, type 2, hepatitis B virus, hepatitis C, HTLV 1 and 2. So those are important to test, and other transmissible agents. Even in autologous, if it is not regulated, it's still being tested by some manufacturers because the product goes to a GMP manufacturing, and those manufacturers, those providers don't want to have something coming contaminating. So even if it may not be regulated, it's a good practice to test it out using CLIA certified labs. Overall goal to have contamination reduction throughout the process. And when we think about final product release testing, you're thinking about making sure you have the right cells with the right viability. The other thing we're going to talk about in this chapter, we did cover a little bit, but we're going to talk more here, is optimizing your cell collection procedure. So as we think about these cells, they are the raw material for the manufacturing. And we always look for, like as a manufacturer, you always look for enough cells so you have less chances or less probability of manufacturing failure due to non-availability of your target cells. So how can we optimize the cell collection procedure? So as we know, we talked about the cell densities. I'm not going to go into too much depth, but when we think about the MNC, and Liz did a great job starting up, that when you're packing these layers, platelets stay on the top, RBC and granulocytes at the bottom, if I'm just taking a cross section there, and then the MNC layer is in the middle, where the specific gravity is close to 1.08. So you're try to targeting this layer with this specific gravity so you can get the maximum cells of the targets of your target choice, right? That's what you're trying to do. But in this case, again, greatly mentioned by Liz, a mobilized donor compared to a non-mobilized donor, they're very different. So you may have to think about how do you optimize your collection to a particular patient population as you're collecting those cells for a particular therapeutic indication. I'm going to head it back to Liz for explaining it more beautifully than me. So Liz, back to you.
Oh, no, thanks, Dalip. You did a great job. So one thing that I wanted to mention that is available to operators to help optimize the procedure to ensure that you're collecting the target cell that you're aiming for and minimizing what we call off-target cells. And really, when I'm talking about off-target cells, I'm talking about red cells, granulocytes, and platelets. That is the use of the collection preference. And I kind of alluded to that earlier. And the collection preference really is talking about how deep in that buffy coat layer, the MNC layer that you have separated out from the whole blood, are you collecting? So operationally, what is happening during the procedure is the operators are looking at this collect line, and that picture that's actually showing an Optia is looking at that thin collect line coming out of the centrifuge. Now, in that example, you can see here is they're kind of monitoring that for the color, with the idea being if the color in that collect line is dark, you know that you're really deep in that buffy coat and you're scooping out a lot of that buffy coat and skimming the top of that red cell layer, moving that up into the collection bag. Now, the inverse is also true. If you were to look at that collect line and it looked yellow like plasma, you didn't see a lot of cells streaming up in that line, you would know that you're collecting high in that buffy coat, more closer to that plasma layer, and potentially contaminating that product with a little bit more plasma, or platelets, because that's where the platelets live during this specific gravity separation. So the operators need to be given that fine-tuned control in order to kind of hit that, what I call the sweet spot. And the sweet spot is really what we say is that salmon color, right? You're avoiding getting too many red cells and granulocytes. And then you're also avoiding that light color that has tends to have a little bit more platelets in it, but you're really targeting that MNC layer. And how they do that is by the use of the collection preference. There's a picture of the screen that is presented to the operator. They use the arrows to dial in a specific number. Now, the number doesn't really mean a whole lot except for a communication tool to that operator. So for this example, if I were to look at that picture, that color is too light. I need to darken it. So what I'm going to do is I'm going to arrow down from 50, and I'm going to let the interface change. And how does that interface change? It changes by the control of the plasma pump. The plasma pump will slow down or speed up based on that fine tuning that the operator is requesting Optia to do. And then that moves that interface either higher or lower in relation to where it is. Okay, so that is one way that they can optimize what is getting that starting material. Next slide, please. Oh, yeah, thank you. That was a video. When we talk about, balancing this purity versus yield, this becomes a really interesting question that we like to talk to developers about. And it's really understanding what is your tolerance for having red cell contamination. I mean, we know we're going to get some red cells up into that collection bag just by default and how things separate in specific gravity separation. But sometimes when we're only talking about purity, okay, that comes to a cost at yield. Well, that might not be a great option too. So it's being able to balance these two and have that understanding that, hey, I need a specific yield in that bag, but I want to try to minimize off-target cells as much as possible. So when we talk about purity, okay, generally speaking, when you want to minimize red cell contamination, you're going to be collecting at a higher collection preference. You're going to be closer to that plasma level, but not to the point where you're letting some of those target cells escape and go back to the patient. Now, when we talk about yield, hey, I just need those cells in that bag. I can handle the red cell contamination. I can handle granulocyte contamination, example. You're going to be maybe collecting a little bit deeper into that buffy coat. But again, this is a conversation because each developer has specific tolerances, and it really needs to involve, like Erin had mentioned, this collaborative effort of us coming together to speak about it. Next slide, please. Sorry, I did that. I kind of already said all of that. So when we also want to talk about common causes of what we mean failure or off-target cell contamination. When I mean failure, I mean that you did not get the cell yield that you anticipated for that particular collection. And there's, 4 big buckets that we really think about when we have to go in and try to investigate, well, what happened to this collection? Like, why were we not able to meet our goal? And this is really a good plug that I want to let people know who are on the webinar is Optia has a hard drive on it that keeps all of the data for that particular procedure, and it stores it. And we're talking about, and people are like, well, what kind of data does Optia keep? It keeps pump flow rates, it keeps alarm states, the images that are captured by the AIM system. Remember, the AIM system is the one that keeps that interface at that collect port. It captures all of those images, any operator changes that are made. And it's really good if there's something going on with your collection that you're not quite understanding. We have a team that comes together that looks at those particular run data files that Optia generates and maybe can help with, suggestions on how we can help get those target cells up into that bag the most efficient as possible. But when we really kind of look at it from an aggregate level, there are really 4 big buckets that really impede the ability to get those target cells up into the collection bag. And the first is what we call an unstable interface. The best example I can provide of an unstable interface is when you have alarm conditions that are preventing. So anytime Optia goes into an alarm state, those pumps that are moving the whole blood and the components of blood around in the kit, they stop. So if you have a specific time frame that you're doing these collections in, say 4 or 5 hours, and you have a lot of pump stops during that procedure, that's going to inhibit your ability to collect that target cell efficiently. That's a good example. Unanswered alarms or alarms that aren't answered appropriately. Sometimes we can see those in the run data files, and we can come in and help operators understand, like, oh, well, maybe you could do this next time, or is just an example. We had already talked about clumping. Proper AC management is a key to ensuring, that you have good flow of blood and the components of blood through that tubing set. If you have micro aggregates that are kind of plugging up those ports, it's gonna cause issues with you to be able to get that target cell into the collection bag. And then lastly, buffy coat accumulation. Don't see that too much in the CAR T space, but if you're doing stem cell collections where the patient has had a really robust mobilization, that can become a problem. And we can also help teach what you need to do if you see that buffy coat accumulation. And next slide. The last slide I'm going to leave you guys with is considering prediction algorithms. Prediction algorithms really take into account the patient characteristics that help you determine how much blood volume based on that patient characteristic do you need to process in order to get that target yield that you're wanting up in that collection bag. Down here below are some examples of where prediction algorithms can be quite advantageous for a collection facility to consider. So we see patient 1, who's a smaller patient, 3-liter total blood volume, maybe not a robust pre-collection cell count, 500 mils per microliter. And this particular site was processing a standard 4 times total blood volume for every patient that they saw. And we can see here that the outcome of their collection procedure was 3 times 10 to the 9th. They missed the target cell yield. Compared to the patient 2, let me go back. What that would mean for that patient 1 is that they would have to potentially come back for another day collection. Prediction algorithms might have helped in this situation where you could calculate how much blood volume you need to process, and maybe they did another total blood volume and they were able to get to that yield for that date, thus preventing the patient from having to come back. Patient 2 is a bigger patient that had maybe a little bit better cell count before coming into collection. They standardly process 4 TBVs, and what we see happen here is they over-collected cells. Now, some people don't see over-collection of cells as an issue, but it can be when you think about storage capacity and who is going to be storing those cells, particularly in the CAR T space. Is it going to be the site that did the collection, or is it going to be the manufacturer? So prediction algorithms are a good way to consider the patient attributes and the effect that it does have on the overall collection process. And that is it. I appreciate your time, and I think we're turning it over now for a Q&A session.
Great, thank you so much, Liz, Erin, and Dalip. We have a couple questions on the Q&A. I'm going to open with one kind of more generic questions, from the ARM perspective, right? We have a lot of our members who are the CAR T developers. So, they're basically the customers of the collection centers, right? So I guess my question is, why is it important for the developers to understand apheresis, and how can developers and collection centers collaborate more to, help overcome, I guess, supply chain issues or any issues that, that come up in order for them to be able to provide these therapies to the patients?
Yeah, I can start and maybe Erin and Liz can chime in. So when you think about the collection centers, when you think about CAR T space, for example, I'll take that as an example. It's a very specific market. They're commercial product. There are a lot of clinical trials going on. So these collection centers, first of all, are resource limited in terms of the collections they are doing. So if you collaborate with them, it's good for them to understand what you're trying to hit so they can work with you much more better rather than just asking them, oh, give us this, and they don't understand. They'll be more involved. That is for sure. And there are like clinical considerations. I'm sure Erin is going to talk about that, but there's like from a collaboration and communication perspective, it's a resourced burdened industry. So having that communication is important. Why it is critical clinically to talk. So I'll pass it to Erin, and then maybe Liz wants to chime in. Yeah.
Oh, definitely, Dalip, you hit the nail on the head. Transparency and open communication is vital. Knowing what your source material, your patient, especially for autologous CAR T, is capable of, what the collection space looks like, and any issues that could potentially come up in that could hinder an effective efficient collection is key for all players on the entire supply chain to understand, to keep that transparency vital. So, if, for example, Liz is collecting my CAR T cells, I have vascular access issues, so there's going to have to be a second day of collection. Those cells going to Dalip for manufacturing, he can know and plan effectively. So I think just making sure we're all on the same page to get that very efficient collection, effective collection, and a very efficient time point so that those cells can be made for that patient is absolutely vital. And understanding the components of the apheresis life cycle where there could potentially be issues is also vital. Liz, I know you have some excellent thoughts as well, from not only being in our space, but standing there at the bedside as well.
Yeah, I think the one thing that I just wanted to add is communication on what that starting material is, like any kind of testing that you're doing. And maybe if you could report back to the apheresis departments. I know I've done personally a couple of cell collections or CAR T collections where it was just, well, you didn't get what you needed, and so we need you to do this patient again. And it was like, well, what really happened? Like, we don't have a complete picture of what happened, so then I can't get better for the next time I see that patient. So I feel like that's a piece that might be missing. We could work on collaboratively in the industry. Absolutely.
Great, thank you so much, all. Let's see, there's a question from the audience. So if you prime your donor blood, if you prime with donor blood, how might that impact potential considerations regarding autologous products? For example, justification for the mitigation of risk or of advantageous agents.
I can
take that.
I can start taking that. And Liz, you can let me know if I'm off beat or not. So this, as a transfusion medicine apheresis physician, when we prime a patient or a donor, we are priming with blood that is matched specifically in the blood bank for them. The majority of the prime blood does go in a waste bag, but there is several tens of mils, potentially more, that could reach the patient, that could potentially reach that collection bag. It is considered like a transfusion. So it is obviously an allogeneic transfusion, but there is not enough of it to change the labeling of that autologous product. That helps answer the question. It would just be like if, for instance, your patient got transfused, then this often happens with CAR T collections in my world, transfused the night before, platelets or red cells, and then we're collecting them the next day. We needed their platelets to be up to a certain level. It does not affect the fact that it is still an autologous product. It is not enough allogeneic blood product to change that. Liz, did I miss anything?
No, I think that's great. And the PAC cells are lymphocyte depleted cells, yeah, too,
which
as
is
well.
Perfect. Yeah, great point. Yep.
Excellent. Thank you. Okay, so I think we have time for maybe two more questions. There's one question about CAR T moving up as first and second line standard of care treatments instead of last line treatments. So how does this movement change, the technical considerations or the patient preparation that you all presented, or if it affects it at all?
For my 2 cents, patients who are earlier in a treatment course means their marrow is potentially in potentially better shape. They have not had all that additional therapy, which is very hard on an entire system. Liz, would you and Dalip, would you guys agree with that?
Yep.
Absolutely.
Yeah,
Absolutely.
Yeah.
Yes.
Yeah.
Hopefully better collections, better collections.
Yeah.
You still have to keep patient safety in front, as Erin does and Liz
Oh,
does all the
God,
time. Like you
yes.
still having less treated patient may help, but again, that safety, that constant supervision during the collection still stays the same, if that's the question. Yeah, it will make maybe collection better, as Erin mentioned, but overall, the focus that Erin gives or Liz gives when we are in the clinic, the focus and the supervision will not be no less. It will be still the same amount of focus for each patient.
Very, very well said, Dr. Sethi. Wonderfully
Thank
said.
you.
Yes.
All right, so last question to wrap up. So I think, we as an industry talk a lot about standardization. So I wanted to get your thoughts on, as we move, there's this trend towards standardized source material. Like what does standardization on apheresis look like in the context of CAR-T? What is already standardized and what can or cannot be standardized?
Yeah, so I may chime in and then we can please chime in. So this was a big topic at ISCT this year because we did a lot of roundtables on apheresis and standardization to clarify, and this is what we said at ISCT too. Standardization doesn't mean same collection from each patient. Standardization could more be, as we are talking about, the data collection, the more about labeling. Each patient needs a different care and it has to be optimized for the patient. We are trying to create more prediction algorithms so the optimization happens much more smoothly. So for a particular therapy, for a particular patient population, you can have the same protocol, but again, within the protocol, there will be variability in terms of when the patient is managed. So I will start there and I'll go to Liz and Erin, whoever wants to chime in.
Yeah, I agree with what you just said, Dalip. I think it's hard to standardize the configuration of Optia because you have to consider the variability in the patient. Like we were kind of showing in that Buffy coat, there's a variability between a mobilized patient and a non-mobilized patient. And even in the non-mobilized patients, there's variability between those patients as well. So I think the specific button presses that happen on Optia really wouldn't be where we could standardize. But to your point, data capture, what are we standardly collecting from data post-procedure, pre-procedure? Are we all getting pre-counts on these patients and know where they're starting at? Would probably be a really good direction because then that would help us maybe standardize those collections a little bit more. But we have to have the data first before we can make those big decisions.
And I think you can always standardize your clear, consistent communication.
That's a good point.
So that your approach to these variable patients is very standard, very transparent, especially how you capture that data for yourself and your patient population so that you can continue to move forward as a big team together to get these patients the treatment that they need.
Yeah, very well said.
Excellent. Well, it is 12:03 currently, so we're a little bit over time, and we have a few more questions that we didn't get to answer. But so we'll send it to you guys, and for you guys to answer offline, and we'll post it in the ASO webinar website. I want to thank you all of them again for taking time out of your busy day to share your expertise with our audience, and thank you for the audience also for engaging with us in the Q&A.
Thank you.
Stay tuned for the next ASO webinar, and
guys.
have a good rest of the day, everyone.
Thank you.
Thank you so much. Thanks, everyone.
Thanks, guys, for your time. Appreciate it.
Bye-bye.